And then, yeah, I will then talk about some of the more personal aspects of how I find doing academic research. So when I talk about senescence, I tend to show this diagram, which is one I made myself. But I think it's also well worth actually showing you what do senescent cells look like. This is just a sort of like idea of what we think they look like. And so these are some images that I've actually taken from cells that I work with in the lab. And so the top image here is just showing what we would consider being healthy cells in a dish. And the bottom image is showing some cells that I've made senescent. And if we look in a bit more detail, what you can notice is that these senescent cells, they don't look particularly happy. You can see all these vesicles, which we'll go into a bit of detail in a second. But you can really see that they have this strange sort of abnormal phenotype, and that's one of the sort of characteristics of senescent cells. So hopefully this actually now gives you some sort of perspective about what we mean when we describe senescence. So, I mean, what actually are senescent cells, and why would a cell become senescent, and how do cells become senescent? So there's actually many different ways in which you can get a cell to enter into the state that we call cellular senescence. And so for many of you in the audience, you might have heard about telomere shortening, which is what happens when a cell divides. And if telomeres get too short, a cell can enter senescence. But that's just one way in which a cell can enter senescence. In particular, my research is mainly focused on DNA damage-induced senescence. There's also oncogene activation can induce senescence, oxidative stress. And essentially, the way I like to view senescence is that it's just a sort of a stress response that the cell undergoes. And it's a way to protect the cell and essentially try and restore the damage. And if it can then go back into a healthy state. And if it's unable to repair the damage, it persists in a senescent state. So essentially what you need to take from this slide is that there are just many different ways in which a cell can become senescent. And so despite there being all these different ways in which a cell can become senescent, there are actually some sort of general characteristics that we can put to senescent cells which help us study them and understand them. So the first kind of very key hallmark of senescent cells is that they enter this irreversible cell cycle arrest. So the idea is that once a cell's become senescent, it sort of stays senescent. There's no way that it can go back and start dividing again. Another thing that you might have noticed from the previous image that I showed is that there's a lot of damage accumulating within these senescent cells. And that's partly a reflection of why they enter senescence in the first place, is because of the damage that caused this process in the first place. And so in the case of DNA damage, or oncogene activation, so these are like genetic mutations. You can view senescence as kind of a tumor suppressive mechanism. So if a cell's damaged, you kind of don't want that to keep on proliferating because it might become tumorigenic. So in many ways, senescence can be seen as a good thing. It's protecting damaged cells from causing more chaos within the body. And so another thing that's really noticeable, and we'll go into a bit more detail about because it's probably most relevant in terms of aging context, it's a secretory phenotype that senescent cells have. That is, they start sort of secreting additional components into their surrounding environment. And that's really important for how the surrounding tissue and the body responds to these senescent cells. So to come back to this image that I showed at the start, what this was meant to be describing is the secretory phenotype that senescent cells have. And all of these sort of, well, it's more noticeable in this image here. Essentially what these small dots that you can see, these are vesicles that are basically packed full of proteins that are going to get secreted out of the cell into the surrounding environment. So what is the point? I've just tried to explain why a cell would enter this irreversible cell cycle arrest, as in it's a protective thing for the cell to do to prevent it from becoming tumorigenic. But what about this secretory phenotype? What are cells actually doing? What are they secreting into the surrounding environment, and what is the impact of that? And so I've just listed here a couple of the different components that tend to make up the secretory phenotype. I'll just sort of go through a couple of them that I think are most important. The key ones to understand are things like cytokines and chemokines. These are basically communicating within the body, and in particular, they're really important for activating the immune system to come in and respond to the damage, which likely caused the cell to become senescent in the first place. So in many ways, these are sort of like signals that the cell's saying, help me, there's something not quite right here, I'm kind of stressed out, there's some sort of damage, we need to repair it. And in particular, that's also why you can see growth factors. In particular, that's been thought to be able to activate stem cells in the surrounding environment to, once the senescent cells get removed, to then replace the tissue and basically restore homeostasis, which I tried to explain yesterday, within the tissue. And so, therefore, it raises the question as, are senescent cells a good or a bad thing? I've tried to explain a couple of reasons why we think they're a good response for the cells to undergo, partly because they are tumor suppressive. It stops, in quotes, bad cells from dividing, but they're also now been shown in studies to be really important for wound healing and being able to activate the immune system, to activate stem cells, to come in and basically restore the previous tissue. The problem arises when these senescent cells aren't removed and they end up persisting in the tissue. And the idea is that you're continuously creating this inflammatory environment, and if you have a senescent cell in a tumorigenic setting, it's actually able to, with these cytokines, these growth factors, to stimulate potential tumorigenic cells to keep on dividing, and essentially aggravates early stage cancer. At least that's currently what we understand. And likewise, in the context of aging, we end up with this chronic inflammaging or inflammaging that you may have heard about. So this is why senescent cells are often regarded as being quite a confusing subject to understand, because they have both these good aspects and these negative aspects. And so how do we, firstly, there's three key things that I therefore want you to understand about senescent cells. Firstly is that they are very heterogeneous. They sometimes seem to be good, sometimes bad, and that's partly a reflection of the main inducers that cause the cell to become senescent in the first place. They're also dynamic. Once a cell's entered senescence, it's now senescent for however long it persists within the tissue, and it's not static, it's very energetic. It keeps on secreting things and communicating with the environment. And it's been shown that the secretory profile that it generates actually changes over time, and it's been very much underexplored at the moment. And yeah, essentially these cells are active. They're often described in like popular media as like zombie cells, but I tend to not like that description, because these really are sort of active cells that actually have some beneficial effects within certain responses, such as wound repair within the body. So what about so-called xenotherapeutics, being able to remove senescent cells from the body? So xenotherapeutics, the way I see it, there are kind of like three subdivisions which people are trying to explore at the moment. The first ones that you've most likely heard about are senolytics, where you actually just try and remove the senescent cells by using various different drugs that are being studied. There's also so-called xenomorphics. What they're purported to do is to be able to blunt the inflammatory aspects of the secretory phenotype in a way to prevent this inflammation. And then the third aspect is so-called immune surveillance. And so that's something that's probably less well understood at the moment, but it's understanding how these senescent cells are communicating with and activating the immune response, and what components of the immune system are actually involved in removing senescent cells from the body. So because there's the most data on it, I'll just briefly go through senolytics. I think this study was actually shown yesterday, if I'm not correct. So there's quite a few studies now that seem to show that senolytics have some potential, and in fact, from these preclinical studies done in mice, they've now entered into early stage human clinical trials. And so just to give some context, in this study, they were giving a combination of drugs purported to be senolytics, dasatinib and quercetin. I'm not sure if you can entirely read that from that slide. And the other thing that's worth mentioning with senescent cells is that this state that they enter, it takes a bit of time for the cell to go from that initially healthy state to some sort of damage, and then to actually establish this irreversible senescent state. And so when I study senescence in the lab, I do this DNA damage-induced senescence. It takes about 7 days for my cells to actually enter into a robust senescent state. And so James Kirkland, who's kind of pioneering a lot of these clinical trials in senolytics, has come across this hit-and-run approach, where actually if you want to develop a therapy, you only have to give it once every so often, because there's this delay between actually removing senescent cells that are already present and then for them to actually accumulate again, because it takes a bit of time for a cell to fully establish senescence. So this is what I was supposed to talk about, which was the theories of aging. But as I said, we had a pretty good discussion about that yesterday. And lo and behold, senescent cells have also been proposed as some sort of theory of aging. And so essentially this diagram is showing that we accumulate these senescent cells of age. exactly why that happens or how that happens is a bit unclear. My current general feeling is that this is somewhat due to defects in the immune system, such that the senescent cells are not being cleared effectively and therefore persist and further aggravate other diseases. But I still think this is being uncovered, and so I'm not going to go into too much more detail than that. So there are some really big questions left in senescence that both the lab that I'm currently in and other labs are actually trying to address. The first one is how exactly these senescent cells are communicating with the immune system, and what components of the immune system, what cells, the T cells or what subtype, are actually involved in removing the senescent cells from the tissue, and potentially could we activate those cells in cases of aging where the senescent cells are not removed. Then where my project probably more fits in is with the decision making. Why does a cell enter senescence in the first place? Understanding basically the underpinning molecular mechanisms that drive a cell into senescence rather than apoptosis, a form of cell death. And then a sort of more general question is, why does cancer risk increase with age, and do senescent cells play a role in that process? And so that somewhat leads me on to talking more about my own PhD. So I'm at Cancer Research UK, hence the last question I mentioned. And so I study senescence, and in particular, I study the role of P53 in senescence. I'm not going to go into too much more detail than that. If you want to ask me exactly what I'm doing, I can tell you at a later time. But I thought now I would just sort of give some perspectives on how I find doing research in academia, and also some of the potential pitfalls that I think maybe you should be aware of. So yeah, just some of my experiences. And I have to say firstly that by and large, this is part of my lab that I'm in. It's been a good experience for my end, but I've obviously surrounded by PhD students and have a general good perspective on how things are going at the moment. So one question I always ask myself is, how can I be a better scientist? Is there something that I can learn from previous scientists, or like what is the best way to do science today, given that we've had all these rapid advances in different tools and just the ability to generate these really large data sets? And so I don't want to stand here and say I'm a great scientist. I'm a scientist in progress. But I think essentially a lot of what it does reflect back to is just having a good understanding of the scientific method. And essentially, to break it down as illustrated here, it often starts with some sort of observation, some question that you notice. So it could be, why does a cell become senescent? Why do we age? But essentially trying to go from those initial questions to actually formulate hypotheses, and in particular, testable hypotheses that using the current equipment that we have, we're actually able to test. So the way that I think is currently the best approach is to initially have very clear answers that come from your questions, such that if it turns out your hypothesis is wrong, you at least fail fast and you can start again. But if your quick tests do look positive, you can then go on to these larger data sets. And then from those observations, you sort of reiterate the process, because your data should therefore lead to new questions, and so on and so on. Now, that said, it's not necessarily trivial to just follow the scientific method. There's always going to be issues that arise. So I decided to use this analogy of a treadmill to sort of illustrate a couple of aspects that I find as pitfalls within my PhD process. And the first thing that I think is quite interesting is about this idea of lab turnover. I've been in the lab now for 4 years, and there's actually been quite a turnover or treadmill effect of people within the lab. And that actually has a massive impact, because a lot of the time you need the more senior members of the lab to actually train and teach and pass on like the wisdom, so to speak, to the younger students. And essentially every time you lose lab members, you also lose information. And that's not just because that person's gone, but also the documentation of their research is also left behind. And I'll come later to give some potential solutions for each of these, so I'll just leave you in suspense for now. So along with the treadmill, I mean, doing a PhD or any position in science is hard work in general, so it's just a lot of work. And then there's a lot of training and learning. You have to work out how to answer your hypotheses, work out what exactly the tool it is you want to use, and you also need to have a good understanding of the different experimental techniques that are available to you, and also the limitations within them. And I find that these days there's just so many different tools that you could use. It's very hard sometimes to know which ones to pick. And I think that there's a massive lack of teaching just in terms of how to generate good experimental design to actually go about testing these hypotheses. And so this isn't just limited to my work in senescence, but I think it extends more broadly to the really big questions in the aging field, and potentially why we still struggle to make big findings. And then there's the fact that it's also a real struggle to keep pace with the science literature. There's tons of papers that come out all of the time. That's somewhat a reflection of the fact that some PhD programs actually have an expectation that you have to publish before you get your PhD. And therefore you end up with a lot of papers that maybe they don't have negative data, but they don't really have positive data either. They just sort of have a little bit of information. And it takes time to read scientific papers, and we're just giving more and more out each day. And it can be very hard to keep up with the science literature. And so I know now that AI is being able to sort of summarize papers in short form, but I think there's massive room for improvement in trying to facilitate the work of a student or any person within a lab by having better modes of communication of actual interesting updates. And then the last point was about analogies and storytelling that happens in science and how that can often misconstrue the actual results. Ironically, I've used my treadmill analogy. So actually, I quite like analogies. Storytelling, it definitely helps, but it's also worth being cautious about this sort of cherry-picking approach that happens when it comes to publications. So I can't quite remember what I was going to say with the slides, but I think the talk was meant to be a reflection of how I currently have a sort of day-to-day view of what my time looks like. So by and large, a lot of the time for a PhD student is spent in the lab actually just doing experiments and collecting data. And the reason why it's so big is because there's somewhat an expectation that we should be in the lab, and also the fact that often maybe we don't design our experiments correctly, and so we have to repeat them, and we end up just collecting all this data and then leaving very little time to actually be analyzing and thinking about our data or planning robust experiments. So actually the bottom approach is what I think is a better way to go about doing a PhD or any sort of scientific research, is to actually dedicate more time to the planning aspect and to analyzing and thinking. And I generally think that is a pitfall in the fact that I think there's some sort of massive expectation that you should just be in the lab all the time. And I think it should be more thoroughly taught, or at least accepted, that it's more effective to have all this time to analyze the data that you're collecting and to think about it and to plan it, because then you're less likely to make mistakes. And therefore, you can actually, just by doing that, reduce the amount of time you have to spend collecting data. And in fact, this is kind of reflected in this discussion piece that Purnas wrote a couple of years ago now, where he kind of sees that as, with all these new genomic approaches, proteomics and transcriptomics, essentially all these different techniques that we use in the lab these days to just generate loads and loads of data, but we don't actually spend as much time analyzing it or actually thinking about it. And sometimes more simple experiments can give us more clear results, and they can lead us to faster failing or faster discovery making. So he took this quote from Sidney Brenner, which is, "We are drowning in a sea of data and starving for knowledge." And so I just think it's something that's worth reflecting on a lot during a PhD or just scientific methods. So this is my general feelings about how we should be thinking about the scientific method and where we can improve it. So having this more time to think, having better training and teaching about the scientific methods, to be aware of the toolkits available and how you can pick the right equipment to answer your experimental question. And by and large, funding will also obviously help, just facilitating the teaching and training aspects. But also, a lot of the time, at least for PhD students, they have a short period of time in which they have to get these papers done. And if you have more funding available, it sort of alleviates some of the stress. And stress also leads to mistakes, and mistakes lead to aberrant results. So that's the general solution. And this is just to illustrate how even addressing the toolkit question is not trivial. This is just illustrating the many different toolkits available with CRISPR, which we can look at it and say, oh, that's great. There's so many different things available for us to use. But actually, from the other side, it's well, which one do I use? Which is the best one to use? Which one's going to give me the right data to answer my question? So to come back to the treadmill, I think in terms of the lab turnover approach and losing members of the lab, really documentation is key. Having better tools to either have digital documentation or just better ways of keeping track of what's being done. So that's my general thoughts. Keeping pace with the science literature, I think there are ways, especially with AI, being able to summarize papers, but also just being able to make papers more digestible, and that's partly could also be alleviated by just having less papers in general and only publishing when you have really big insights. The hard work, I guess, you can alleviate by just having a more supportive environment. And then in regards to choosing the right tool, I think it's really just about education and better teaching and training. So I'm not sure entirely if that's what Kat was wanting from this session, but hopefully this has given you an insight into Senescence, but also some sort of insight into what it's like to do research as a PhD student. And yeah, happy to talk about questions now, just come find me later.